Hello, my name is Riley Rafferty, and I am presenting this on March 15th, 2026. Today I will explain how to solve a 3-point gene mapping problem. Gene mapping is used to determine the relative positions of genes on a chromosome using recombination frequencies. A 3-point test cross analyzes 3 genes at the same time, so we can determine gene order, calculate map distances, and analyze crossover events such as interference. The first step is organizing the offspring data into a table using the genotypes and the total number of offspring. After adding all the counts together, the total number of offspring is 5,742. This total will be used later to calculate recombination frequencies. Next, we identify the parental or non-recombinant classes. These are the 2 genotypes with the largest numbers, because they represent the original chromosome combinations. In this dataset, the parental classes are ABC with a total of 1,362 and plus with a total of 1,340. Now we identify the double recombinant classes. These are the smallest numbers in the data because double crossover events are rare. The double recombinants are A++ with a count of 100 total offspring and plus BC with a count of 140 total offspring. Together they give a total of 240 double crossover offspring. To determine the gene order, we compare the parental genotypes to the double recombinant genotypes. When comparing these combinations, gene A changes while the other genes remain the same. This tells us that gene A is the middle gene, and the correct gene order is BAC. To calculate the distance between B and A, we add the recombinant classes plus B plus and A plus C, along with the double recombinants. This gives 1,060 recombinants. You divide this number by the total offspring. The total offspring is 5,742. Dividing them gives you about 18.5% recombination. So the distance between A and B is about 18.5 centimorgans. We repeat the same process for genes A and C. Adding the recombinant classes and double recombinants gives 2,220 recombinants. Dividing by the total number of offspring gives about 38.7% recombinants. So the distance between A and C is about 38.7 centimorgans. Next, we calculate the expected number of double crossovers by multiplying the recombination frequencies. This gives about 411 expected double crossovers. The actual number observed was 240. The coefficient of confidence is 240 divided by 411. This equals 0.584. Interference is 1 minus 0.584. This means that the interference is about 41.6%. So about 42% of expected double crossovers were prevented. In conclusion, the final gene order is BAC. The map distance is 18.5 centimorgans between B and A, and 38.7 centimorgans between A and C. The coefficient of confidence is 0.584, and the interference is 0.416. This shows that crossover events can influence each other. These calculations allow geneticists to construct maps and better understand chromosome behavior during meiosis.